Hi, everybody. Welcome to this second session of the CCAM Technology and Innovations CTIS webinar series. We held the first session in August on the Biotechnology Ignition Grant to great response from all of you. With the CTIS number 2 webinar, we bring you a closer look at the CCAM technology platforms portfolio, specifically our multi-omics facilities. Most of you are aware that Center for Cellular and Molecular Platforms, or CCAMP, is a part of the Bangalore Life Sciences Cluster, or BLISC. We are one of India's most exciting innovation hubs where we promote cutting edge research and innovation in the life sciences. What you may not know, though, is that CCAMP was once instituted with the main aim to provide access to state of the art technology platforms housed across the cluster. Today, CCAMP is associated with 425 plus academic institutes, established biotech or biopharma companies, research organizations, startups, and individual innovators across India and the globe. We have supported over 180 publications. Our genomics, metabolomics, proteomics, lipidomics, and glycomics facilities together form one of India's best in class multi-omics facilities. With that, I now pass on the mic to my colleagues, Dr. Atif Alam and Dr. Sheetal Kalme from the CCAM Technology Platform Program. Joining them in the conversation will be Dr. Avdesh Pandit from Genomics, Dr. Chaya Patole from Proteomics, Padma Ramakrishnan heading our Metabolomics facility, and Dr. Sabyasachi Haldar heading our Biologics Characterization facility. Over to you, Atif.
Thank you, Debarshini, and good morning, all. It's a pleasure to be talking to you this morning, and I hope we will have a productive, very interactive session. The idea here is to ask questions to the experts who have kindly agreed to give their time for the next hour or so. Just to make it a little fluid, I have put together a couple of slides as a prop to help discussion, and I'll very quickly go through the slides to give you an overview of what we have. Many of you might know it, but for those who would be seeing it first time or some updates from our side, I'll be showing them very quickly. As Chandrakant and Debashini told, feel free to ask questions. You can ask questions when the experts are speaking, or you can ask hold on to them for the later part. So before we dive deep into the technical discussions, another very quick overview of what we do. We do our activities broadly in two verticals, that is technology resources, of which a subset we will discuss today. And then we also work very extensively with life science entrepreneurship ecosystem. And in this one slide, I am trying to do justice to the amount of work we do with the startups, right from the side of outreach to all the way to the acceleration of startups. With this, we have worked with over 250 startups and of various stages. Debashini, correct my numbers anyway. So this is, though not today's topic, I just wanted to give a glimpse in case any one of you is interested to start your own venture, you know where to go. Okay, so coming to the technology resources, again, it is very difficult to condense the amount of resources that we have on the cluster, which is that the three of the organizations together. I'll come to the multiomics platforms a little later. But here I would like to introduce my colleague, Dr. Sheetal Kalme, who very ably manages the multiomics platforms, all the projects, the techno-commercial side of it. While you hear from the technical experts, bear in mind that she is also around to answer any of the questions which are techno-commercial in nature. Okay, so while this is a snapshot, feel free to click snapshots in case you wish to come back to us with any specific questions. Right now, this slide shows the breadth of activities that we do. Quickly, I'll go over with a overview of the flow cytometry facility, where we offer a bunch of services which can appeal to both the sorting and the analysis kind of applications, right from apoptosis, autophagy, cell cycle, and so on and so forth. These are the assays that we have established in the facility and a bunch of flow cytometers which are fairly state of the art, and we are constantly trying to upgrade our infrastructure. Imaging facility, again, this is one slide in which I am trying to compress the capabilities spread across actually 3 sub facilities. That is the atomic force microscopy, confocal microscopy, electron microscopy, and the micro CT facility. Uh, so again, we will have at a later point a separate session for each one of these technologies with the expert in, but feel free to reach out to us in case you have any queries about this. Similarly, the molecular and structural characterization is strongly supported by the most recent and most powerful magnets and the nuclear magnetic resonance spectroscopy, X-ray facility, which has crystallography and small angle scattering machines, and the country's only 300 kilowatt Titan Krios cryo-electron microscope is also available on the campus and is accessible. In fact, there are a couple of very exciting publications that came from users who were not from the campus. The external users used the facility to get some really beautiful structures, and they were able to publish it in likes of Nature Communication and so on. This is the only campus which offers transgenic animal facilities. We have animal models on which we offer transgenic services using CRISPR-Cas9 technology as well as the older traditional technologies. We have the mouse genome engineering facility and the animal care facility, along with the transgenic Drosophila facility. This is where we work with the researchers to develop new strains of uh, transgenic animals. Core equipment is also available in case you wish to do some kind of a pilot scale fermentation or a small scale purification, some analytical work. This is also available. And then something which is in the devices space, that is the microfluidics and microfabrication and rapid prototyping facility. Bunch of exciting stuff is coming up right now. As we speak, we are building this lab, and hopefully in the next couple of months, we would be able to support right from design of a micro device all the way to pilot scale, which would be a capacity of around 5,000 units a month. So that's the goal with which we are working towards this prototyping facility. I am really pleased to say that over the last many years, people have been very kind to acknowledge us or co-author publications arising out of the work that was carried out at the life science cluster facilities. And it has been listed here on this website as a repository. Feel free to go up, check out what has been already published, reach out to us in case you need the preprint or you want to discuss around a similar or something which has been published in a high ranking journal. Very important aspect of this whole resource is that people are trained on these technologies. As you would know, these would be highly complex data sets that would be thrown out by the equipment, and it requires a lot of analysis, lot of expertise. Experts are there to acquire it properly. They are there to help you analyze at some point, but then at some point you have to go back and work in your own lab. And that's where training on these resources comes in handy when you're trained into seeing how to put up a good sample into an equipment, get a set of data which is proper, and you have the proper controls and the standards and so on and so forth, and then how to go back and analyze. To this end, we actually organize training courses almost on a frequency of every fortnight. So have a look at this website, Technology Trainings, or on the Blisk and CCamp social media handles for the upcoming training programs. With this, I'll actually dive into today's topic, and that is the multi-omics platforms, which are I've highlighted in the yellow box here: genomics and the mass spectrometry facility. You can use them as separate or in tandem to get enormous amount of data to try and understand the life and living cells itself. This is where I would like to introduce and have my colleagues take over one by one. share their few thoughts and then maybe take questions. At this point, I'll invite Dr. Avdesh Pandit, who is the in charge of the genomics facility. Avdesh has his PhD from in plant sciences from IAR and has a bit of a has many years of experience in the genomic space and has been with us for last 3.5 years. Avdesh, it's over to you.
Thank you, Atif. I'm audible. Hello.
If you could speak a little louder, that'll be good.
Now. Now.
Yeah.
Yes, yes.
Yeah.
Yes, please. That's good.
Okay. Okay. Thank you, Atif. So today I'm going to give a brief overview about next generation genomics facility. So basically we have 4 sequencing platform in the facility. First one we have from Thermo Scientific company. The platform name is ABI 3730. This platform is basically based on dideoxy chain termination method or the Sanger sequencing based method. We can generate up to 800 base pair good quality sequence read length. It's a 48 capillary based sequencer, so we can sequence 48 samples in one run, and it takes around 2 to 3 hours. We are using this platform for PCR product sequencing, plasmid sequencing, and the genotyping applications. We have 2 next generation sequencing platform from Illumina company like MySeq and the HiSeq 2500. Both the sequencing platform are based on sequence by synthesis method. In MySeq, we can generate the maximum read length of 600 base pair. Uh, there are also different read length kits are available, like the single read 50 base pair, pair in 250 base pair, pair in 150 base pair, and the pair in 250 base pair kits, and the read lengths are available. So basically, suppose if we want to do the microRNA sequencing, and we know that the microRNA are 20 to 25 base pair or 30 base pair long, so we can choose for a single read 50 base pair sequencing, because just we need a 30 base pair sequencing. Similarly, suppose if you want to do the whole genome sequencing, then we need longer read length, so we can choose for pair in 250 base pair or pair in 300 base pair read length runs. So we can generate up to 50 million reads using the MySeq platform, and it takes 48 hours time. This platform we are using for a small whole genome sequencing, like the microbe and the viral genome sequencing. This platform is basically it's good for 16S metagenomics work. Basically, this is the one of the best platform to go ahead for 16S metagenomics work. And we are using this platform for targeted gene sequencing, like the amplicon and the gene panel sequencing. In another platform, the HiSeq 2500, there are 2 run modes are available, the rapid run mode and the high throughput mode. In other term, I can say the rapid run mode is also a low throughput mode. In rapid run mode, there are also different read length kits are available, like the single read 50 base pair, single read 100 base pair, pair in 100 base pair, or pair in 250 base pair. We can generate up to 600 million reads using the rapid run mode, and it takes around 7 to 60 hours. Similarly, in high throughput mode, we can generate up to 4,000 million reads in a run. In other term, we can say 500 in the form of GB. We can generate 500 GB data in one run, and it takes around 29 hours to 6 days. So we can do almost all kinds of NGS application using the HiSeq 2500 platform, like the large and a small whole genome sequencing, human, animal, plant, virus, and the micro genome sequencing, whole transcriptome sequencing, chip sequencing, exome sequencing, or shotgun metagenomics work. We have another platform from Nanopore, and the platform name is MinION. Basically, this platform is based on electric current based detection method. We can generate up to 3 to 200 kb. Recently, some paper have reported they got around 1 MB reads also. We can generate up to 200 million reads using the MinION platform, and it takes around 48 hours. So this platform is we are using for like the MinION, we are using for a small whole genome sequencing like the microbe and the viral genome sequencing, and also for the 16S metagenomics work. So broadly, the next generation sequencing is performed in 4 major steps with the Illumina sequencing platform. First one is the library preparation or the sample preparation. It takes 2 to 3 days. The time here depends upon the type of library. Suppose if we are preparing the library for mRNA sequencing, it generally takes 1.5 days, whereas that exome and that microRNA library prep takes 2 to 3 days time. So after that library preparation, the next step is the cluster generation. So high throughput flow cell takes 3 to 4 hours for the cluster generation, whereas the low throughput flow cell or the rapid run flow cell takes 2 to 3 hours. After the cluster generation, we load the cluster flow cells in the sequencing platform, and it takes 2 to 6 days. And the next step, then we'll get the raw data, and then the next step is the data analysis. It takes days to months. It's the data analysis. It depends upon the time. Depends upon the basically its objective of the project. So at present, we are not providing supports on the data analysis part. Uh, Uh, so these are the applications available with our next generation genomics facility. First one is the RNA sequencing. So we are providing support. We are basically using 2 methods for RNA sequencing: poly A selection based method for almost all kinds of eukaryotic mRNA sequencing work, and rRNA depletion based method we are using only for human, mouse, and rat. We generally need 500 to 800 nanogram total RNA, and the RNA integrity should be more than 7. And the suggested sequencing format is like user can go for single read sequencing as well as the parent sequencing work. The other application, we are providing the microRNA sequencing. Generally, we need 1 to 2 microgram total RNA for microRNA library preparation, and the suggested sequencing format is 1x50 base pair. The another application we are providing the whole genome DNA sequencing. We need around 100 to 300 nanogram DNA for eukaryotic genome sequencing. For prokaryotic genome or the viral genome sequencing, we need just 4 to 5 nanogram genomic DNA. And we can go for it's recommended to go for parent sequencing, but for if it's the genome size is very small, like the viral genome sequencing, we can go for even for single read 50 base pair sequencing also. For chip sequencing, we generally need 5 to 10 nanogram chip enriched DNA for the library preparation, and user can choose go for single read or the parent sequencing work. For whole exome sequencing, we need around 100 to 300 nanogram genomic DNA, and the suggested sequencing read length is parent 100 base pair or parent 75 base pair sequencing format. For 16S metagenomic work, we need around 5 to 10 nanogram genomic DNA. And for V3 and V4 region, we need 200 to 300. For V3 and V4 region, the suggested sequencing read length is parent 300 base pair. And we provide data in FASTQ format. So this is the whole about the next generation genomics facility. I am happy to answer the question if anybody has.
So I'll have a question to moderators. Do we take the questions now, or we hold on till the? Because there's a question on genomics for Avdesh.
Yeah, just.
Debashini?
Yeah. Hi, surely.
Can we take the question?
Yes. So Sachin Kumar is asking that he would like to go for ancient DNA sequencing. What is the best way to do that?
So we, if it's a whole genome sequencing, we can use the Illumina platform for this ancient DNA sequencing work. Yes. What about the other question? Hello.
The other question is from Azim Khan, who is asking what is FASTQ?
Well, FASTQ basically it's a compressed file, and it's also appended with the quality score. So if you run the FASTQ file in any of the software, you'll get to know about the sequence as well as the quality score of each and every sequences.
Okay.
Yeah.
I think if there are, okay, there is one more question. RRBS sequencing for epigenetics can be run using a HiSeq system?
Yes. Yes.
Okay. Yeah, so we will share contact details of all the resource persons once the talk gets over.
Let's limit to 5 questions between the conversation, and then we can take them later on if you could hold.
Yeah, exactly.
I think you can ask 2 more questions if there are on genomics for Avdesh.
Yeah, so there is somebody asking what is the, one second. I am working on fecal samples. Is it possible to generate whole genome sequencing data? From Vinod Shanmuganatham.
Yes, it is possible.
Okay, from Dr. Maitri R, what is the best platform for organelle genome? Best platform for organelle genome?
We can use Illumina or as well as Nanopore also.
Next, shall we get exact quantity of each microRNA after sequencing?
Yes.
Okay, then compared to microarray sequencing, how is NGS different?
Sorry?
How compared to microarray sequencing, how is NGS different from microarray sequencing?
Okay. So the main difference is that in NGS we can get the novel transcript expression also. Whereas in the microarray only we can able to know about whatever the probes are available or the genes available on the microchips. So only we'll get to know about the expression of the genes available on the chip. Whereas in the NGS we can get even the novel genes and the novel microRNA expression as well.
Okay, I think there are a few more questions. Shall we answer them later, or Atif, should we go ahead with? There are a few more questions for Avdesh actually. Atif?
If you just a minute. Yeah, if you don't mind, let's move on. The other speakers are in line, and then request to the participants to please hold on and just bear with us. And maybe they have a bunch of questions. So we'll move to the mass spectrometry aspect of it. Here we have a very large group of experts who basically dabble into multi-omics, literally all the multi-omics from proteomics, lipidomics, metabolomics, glycomics, and a special field of biologics characterization, which we will come to when Sabya is just about to start. So I'll introduce Chhaya, who is the in charge at the proteomics and the lipidomics facility. Chhaya did her PhD in biochemistry from University of Reading in the United Kingdom, and was working with Bear Street Analytics Private Limited before joining us. Over to you, Chhaya. Her expertise is proteomics.
Thank you, Atif, and welcome everyone. So I am a facility in charge of proteomics. So let's start with what is a proteomics. Proteomics is a study of a protein like expressed in response to a stimulus at a particular tissue at a particular time point. So studying these type of protein in depth called as a proteomics. So in proteomics, normal workflow is like you have a control and you have a treatment that you want to compare with each other. So that called as a label-free or label quantification technique. So label-free, what we do normally workflow is we extract the protein from the organism that we are interested in. Extract it, and then you digest the protein with a different enzymes are available that you can digest it. But normally in proteomics, we use lysine C or trypsin enzyme. So once you digest your protein, it gives you a small chunk of peptides. So once you have a small chunk of peptide, you clean the peptide, and then you run on the LCMS. So once you run on the LCMS, you take the data out and you do all the data analysis thing to know what type of proteins are expressed in your sample. So when we do proteomics, there are few things that we have to be very careful about. Is like major is your sample preparation. So in mass spec, we have this say, if you put junk in the mass spec, you'll get the junk. Okay. So that's why your sample preparation sample has to be very clean. So mass spec, the thing is that, hello. Okay. So that's why your sample preparation sample has to be very. Sorry, I think I'm not able to hear you. I think your sentence is breaking in between. Please, can you repeat?
Yeah, I don't think there is any question right now.
I think someone was speaking, right?
I don't think anyone was speaking. Please continue.
Okay, sorry. I thought I heard someone.
No. That's fine.
Sorry. So in proteomics, your sample has to be very clean. Okay, so mass spec is not compatible with the detergent and the salt. During the protein extraction, we do use NP40, tris HCl, sodium chloride, or SDS. So these type of detergent and salts are not compatible with the mass spec. So you need to make sure to clean them up before you put into the mass spec. So that is the thing you have to take careful. For example, if you're running gel, okay, so when you run a gel, most of the time it's a keratin contamination that we normally see in the samples that we get. So preparation of your gels are very critical, and you have to prepare your sample very in a clean environment. So that is the overflow of your sample preparation. So let's, I give you a brief about what type of services we offered here. So we do label-free quantification and label quantification. Label-free quantification is like, for example, you have a control set of samples and you have a treatment that you want to compare with each other. So in that case, what you will do, you will extract the protein from both the sets, digest it, clean up, and then run on the mass spec. And then you take the data out and then you compare what type of protein expressed in your treatment and what type of protein are expressed in your control. So you compare relative comparison you do. In label quantification is the same thing, but what we are doing is we are labeling the protein. Okay, so there are different type of techniques that is available. It's called SciLike, iTrack, and TMT. So you can use this and label your protein, and then you proceed to do your trypsin digestion and your running on the LCMS. Let's come on the intact mass measurement. For example, there are people who are not interested in bunch of the proteins. Okay, they are only interested into a particular purified protein and their mass. What is the exact molecular weight of the protein? So we give that service as well. Reaction kinetic measurement at protein level. For example, if you are only seeing the enzyme and your protein reaction, okay, and you want to see at a particular time point how your enzymes is modifying your other protein. So that type of kinetic study also you can do it using the mass spec techniques. Next is de novo sequencing and N and C terminal sequencing. De novo sequencing is a type of where you use different kind of enzyme combinations to know the sequence of your protein. For example, if you think that your protein is novel and you want to at least try to get as much as sequence of your protein. The one drawback of here is like, because you don't know the sequence of your protein, So it depends on hitting in the dark. You use different combination of enzyme and you try to align it. So in this way, sometimes you get 40% of the coverage or 50. So it depends on uh, the result you get out of it. Disulfide bridge analysis. Disulfide is like, for example, there are disulfide linkages in the protein. Okay, you purify the protein and you want to study what cysteines are having a like forming a bond with each other. So that kind of service also we provide. And protein separation based on the PI. If you want to say, we have an off-shelf fractionator which fractionate the protein based on that isoelectric point. If you have some isomeric proteins that you want to separate it out, so you can separate here. Next is protein-protein interaction. It's called interactome studies. For example, you are using one of the protein, okay, and you want to know what other type of protein are interacting with your main protein. So what you do is you do immunoprecipitation, pull down all the protein that going to interact with your protein. You study those interactomes. Next is post-translation modification. Post-translation modification, like right now we offer phosphoproteomics, ubiquitination, and acetylation. So they are the modification on your proteins. So specifically, if you want, you can study those modification and see how these modification are affecting your signaling pathways. The procedure to contact to us is you can contact to my email address or service@ccamp.res.in. The step that we follow is once you register on the CCAMP portal, we have a telephonic discussion with you, understand what is your requirement, what is your experimental workflow. And once we approve on the experimental plan, then you submit a sample to us. And then we follow it up from sample processing to result report uploaded. And we try to give the results in 12 to 15 working days. Can we go on the next slide, please? Thank you. So in our facility, we have two instruments that it's first is Thermo Orbitrap Fusion Tribert. That is the latest addition in our facility. And this instrument we use highly, it's coupled with the nano LC system, and we use extensively to do in-depth proteomics. Other is Orbitrap Excel. It's also a thermo scientific instrument, and we use it mostly for doing intact mass measurement or protein kinetic studies, these type of experiments. So coming up with the software that we have in-house to do proteomics analysis, we have Mascot Distiller, we have Thermoproteome Discoverer, we have Peaks 8, and we do have a MaxQuant and Perseus. We do this software as well. Yeah. Can you please?
Sorry. Chaya, since you are also looking at the lipidomics workflows, can you just quickly share a few lines about it, or you would? I know you're not prepared with the slides, but
yeah, I'm not prepared with the slide because it was proteomics. Okay, so for lipidomics, I will give you a brief overview of what we have in the facility. So we do give complete lipid profiling, and we do have a targeted. If you are only looking at only particular species of lipid and you're not interested in anything else, so we do have a targeted profiling of the lipid species. So in campus, we have QTRAP 6500 and QTRAP 5500 from SciEx that we extensively use for MRM-based lipid profiling studies. If you have any question about lipid, I think we can take it like after this session.
Offline, yeah. Sure. Thanks, Chaya. Debashini, are there any quick questions for Chaya right now, or we move on? Debashini?
I think I can, sorry.
Yeah, Chaya, there is a question from Shweta Jha. She's asking, do you provide protein identification results or just raw data and peak list?
No, I think we do give our, there are two type of data analysis that we provide. We provide basic data analysis where only you get protein identification, like what type of protein you detected. Other type of data analysis, what we provide is in-depth data analysis, where we do give a comparative study, what are the protein are upregulating as compared to control and downregulating as compared to control. So it's called relative quantification also we provide. So based on your requirement, we provide the results to you.
Then there is a question on, do you provide from an anonymous attendee, do you provide DIA LFQ service on Lumos? If yes, fixed DIA, variable DIA or boxcar?
Can you repeat that question, please?
Yeah. So
display the questions, Debashini.
Yeah. So the questions are. So there is a question. I would have put that person on speaker, but he has put himself as anonymous attendee. So the question is, do you provide DIA LFQ service on Lumos, or if yes, fixed DIA, variable DIA, or boxcar?
Okay, I think we don't give DIA services. I think we don't provide DIA services, I think.
Okay. There is Rajiv Kumar who is asking, could you please elaborate a bit about the protein disulfide analysis?
Okay, protein disulfide analysis is, for example, you have take it as a BSA protein, okay? Imagine there are 5 cysteines, just for an example, and 4 cysteines are involved in forming a disulfide linkage, okay? And you want to know what cysteines are forming that bond. Okay, for example, C12 is forming with a C96, or C12 is forming disulfide linkage with 110 cysteine. So that type of information you can get from disulfide analysis. Is that answer your question, Rajiv?
Rajiv, yeah, maybe I can. 1 second. Okay, he doesn't seem to be online.
Okay, so I think next question, I think someone is asking about lipidomics.
Yeah,
I think lipidomics of, yeah, so it's a Prabhavati Devan. I think it's a lipidomics of exosomes from stem cells. You can do it. The only thing is, like, we need to extract exosomes, and then we have to extract the lipid from exosomes. So I guess there will be literature available to you can get the extraction protocol from there. So once you have a lipid, the only thing is that what you need to do is, what we do here is, like, before we do start extraction, we do spike internal standard. We do spike lipid standards, and then we follow the extraction of the lipids.
Okay, there is from Mukesh Dhanka, what is isoelectric point and is it related to charge of amino acids?
No, it's not related. It's, I think, kind of related to charge of the amino acid. Like when you run on the off gel, it's like it's a point where the charge of the amino acid is a neutral. Okay, so that called sorry.
I think this is slightly more difficult, longish answer. You'll have to tell about the strip on which the protein moves and stops and then elutes. So this one you can take offline, I think.
Sure, fine.
This question, can you, yeah, this is not directly mass spec. It'll.
Yeah, there is a general question, and this, I think, can we install the analytic softwares in personal laptops or tablets?
I think if you're asking about the proteomics software, most of the software that I told you about, they are the paid software. So, and they need a quite of very good computer to run, because you are running like 2 gigabytes of files. Like a single file that comes out of the mass spec is 2 gigabytes. So you need a good configuration PC to do that. But there are one freeware software, and it's very people have used and reported in publication extensively. It's called MaxQuant. So MaxQuant you can install in your personal laptop, but you need to make sure that you have enough C drive and space to run those softwares.
Then another.
Chaya, maybe I'll ask here. Sorry, Debashini, if I can interject. Chaya, are there any online tools that people can use? Say you gave them the raw data and you also give them a bit of analyzed files. Then do they have any online tools to use the data and upload there and do a bit of a basic level of
Actually, the thing is that raw file that comes to you, you can't open without the company software. For example, if you are running the sample on the like thermo instrument, so they provide Excalibur software, and you can only see your raw data using the Excalibur software. So there is no software that is available to
so
open the.
So basically it's a instrument logged kind of data. So
Yeah,
since I have a bit of experience with imaging, imaging has a software called ImageJ, which is freely available and developed by, I think, NCBI. So there's nothing equivalent to this in mass spec world.
No, I think
I mean, this is a
as per
question to you
my
and
understanding, we don't have. But what you can do is most of these softwares, okay, most of these software, they give you around 60 days free trial version. So you can install that and you can easily check your raw files.
Okay. Thanks, Shaya. Sorry, Debashini, there's somebody else asking question. I thought this was a relevant, so I just.
Maybe we will keep this the last one for Chaya and then move on. When we want to do a platelet proteomics study, should we follow SciLAC, iTrack, or TMT? Which is the ideal one? Chaya?
Sorry, please can you repeat that?
Yeah. For a platelet proteomics study, we should follow SciLAC, iTrack, or TMT. What sort of analysis is the ideal one?
For platelet proteomics.
Yes.
Okay, so platelet proteomics, you can see iTrack and TMT both are same. Okay, the only benefit you get in TMT and iTrack is the number of samples you can analyze at one go. For example, if you're using iTrack, okay, it comes as 4 labels. Okay, 4 and 8 labels actually. What iTrack kit you use based on that. So, for example, if you're using a kit which has 4 labels, so you can label 4 samples at one point. For example, you take 4 patients, so you can label 4 patients with a different labels. Okay, and then what you do is you extract it, and then after extraction you do labeling, and then you combine your all 4 patient protein like peptide from 1:1:1:1 ratio. Once you mix it together, then you clean it up and run on the mass spec. So that is normally how labeling works. So you can choose anything from iTrack to TMT, both. Both are very well established protocols.
Okay, at this point, I would like to just for the last question for Chaya, Vanita Prasad, I would allow her to talk so that she can ask the question live.
Okay.
Hi, Vanita, will you please ask your question?
Is she on mute?
I am asking her to unmute. Vanita? Yeah, she seems to be on mute. Yeah, she seems to be on mute, so we will follow, we'll go up. Do you, Chaya, the last question is for you, before we move on, do you provide targeted lipid quantification of cardiolipins on 6,500 Q-trap?
Yes, I think we do provide it. But if you're saying targeted, then do you have a specific lipid species that you want to focus? If you can provide us a list of the species lipids that you want to focus, we can set a method for you.
Okay. So a lot of, I think I would like to make a request. A lot of you are asking questions as anonymous. It becomes a little difficult for us to make you, give you the floor. So please try to give us your names. I think with that, Atif, shall we move on?
Okay, sure. Now we move on to the world of small molecules, and which is collectively called metabolomics. And to give insights on that, I'll request Padma to join us. Padma did her master's in biophysics from All India Institute of Medical Sciences, New Delhi, and then worked at Panacea Biotech for a couple of years, focusing on synthesis and characterization of peptide therapeutics. She's the head of our metabolomics facility with us for many years now. Padma, over to you and very warm welcome.
Thank you, Atif, for the nice introduction. So, okay, I'm seeing that black box still there, but thankfully it doesn't affect the figures. So metabolomics, I think it's by the name itself, it is evident that it is, we are talking about small molecules. The figure below is self-explanatory, and you can just see, where the metabolome comes in the scheme of things. And it makes even more sense when you talk in terms of different compartments. So you could talk of the total cell metabolome, or say the urinary metabolome, or the serum metabolomes, and so on and so forth. So that is what the objective is. So the key thing here is that it is a technology which allows you to simultaneously study many molecules at the same time. So that is the beauty of it. And the point is that we have that kind of technology that allows you to do so. And what are the main considerations here? That what are the key challenges in doing so? That although you would like to analyze them in the same method, what would be the cases where that could become difficult? Is that the metabolome is very, very diverse. So, they have different, because of their chemical nature, they can have different polarities. Something can be non-polar, something can be polar, they'll have solubility issues, then they will have something might be acidic, basic, and so on and so forth. Like, something might be volatile, and stability issues. Also, like in other biomolecules, here also there is a difference in the order of magnitude in which they are present. For example, some molecules, like say the amino acids, might be abundantly present. But if you look at some signaling molecules, or if you look at nucleotides or things like that, they will be in a much smaller amount. So can you use the same sample to analyze all of this in a single run, or do you need to concentrate one? Do you need to run them separately? So those are all the considerations that you have to keep in mind. And then it's highly dynamic and time dependent. It's more or less basically extrapolation of the above features. And simple things like when you harvest your sample and how you store it, everything makes a difference to what results you will get. So the common methods that are used are LCMS, GCMS, and NMR. In the next slides, I'm going to be talking more about the LCMS part, but the same thing can be used with GCMS as well. And NMR, of course, I think the main limitation there is the amount of samples. So LCMS and even GCMS undoubtedly are more sensitive than NMR, right? So next slide, please. So broadly, there are two workflows. One we call the targeted, and one is the untargeted. So what we do in the targeted is that you basically do the sample prep. Now, the sample prep, again, now it becomes, since it is targeted, that means you're looking at specific compounds. So the benefit is that you can fine tune your sample preparation. You might not do a simple, say, a methanolic extraction or something. There will be, you can find ways to concentrate it on some kind of, say, an ion exchange cartridge or a reverse phase cartridge or something like that. Or you could use derivatization. So many things you can do when you're looking at specific things. So you do the sample prep, then you run it through the LCMS instrument. And typically for the targeted, the analytical methodology uses called multiple reaction monitoring. And the main thing is that you need standards here. You basically know the parent mass and you fragment it to get the product ions. That becomes like the fingerprint of each compound. And that can be built into a big method. So basically you run the method and then you can see the chromatogram. So this is an LC-SRM chromatogram. So if you see, there are so many channels here. Each channel is corresponding to one metabolite. So in this example, we have shown actually some phenolics, plant phenolics and flavonoids. There are about maybe 22 of them analyzed in a single method. And with the technology or the instrumentation we have, you can increase this number to, like as many standards as you can arrange for. Basically, it boils down to that. And the instrument that we have in our lab is called a triple quadrupole. Traditionally, that's considered as the gold standard for the MRM experiments. So when it is targeted, you can always do a quantitative analysis. So basically, this run is done for the standards. You can use serially diluted standards and you can build like a calibration curve for each of the 20 compounds. If you have 100 compounds, you have to do it all over again for 100 metabolites. And from the sample, you can basically extrapolate and quantify it. So it's fairly straightforward, the workflow, but the challenge, I would say the rate limiting thing here is the method development. Sometimes you have to optimize the extraction and what columns you will use. And sometimes you will see the ionization and all may not be straightforward. So you may have to spend some time. But once the method is established, you can do this experiment n number of times. And some of the like ready to use panels that we have currently are, for example, the phenolics and flavonoids. We have for bile acids and we have for neurotransmitters and amino acids. So this is again custom built. So based on whatever the client's requirements are, we can work on building a method. Then the next one, which is, next slide, please. Next one is the untargeted one, which is what is kind of, catching the fancy of people lately. There are several issues with it, but a lot of work is going on. So here also, as you can see, the first three steps are almost similar. Now the sample prep, it doesn't remain very specific. Usually the goal is to, extract as many metabolites as possible. So generally it would be a more generic kind of sample preparation. But again, one has to keep in mind that, your results can vary based on what kind of extraction you use. For example, I might do 100% methanol extraction, I'll get one result. If I add, make it aqueous methanol, or if I add a little bit of acid to it, so that could change my results. Maybe not 100%, but that will definitely make some difference. So as you would imagine, there would be a million combinations of doing this extraction, but practically you will probably, identify 2 or 3 key things. Basically, for the small molecule workflow, your key considerations are that you have to remove the protein, and that to make it compatible with the MS, you should not have any non-volatile salts. So those are the key requirements. So here also, then you put it on the mass spec. Now the mass spec here, the instrument itself is a little slightly advanced technology. As you can see, we would be using what is called the high resolution mass spectrometry. So these instruments typically have higher resolving power, and the output that you get has higher mass accuracy. So why this is required is because you're trying to identify masses accurately, try to put an elemental composition on them, try to establish their molecular formula, and also search it against external databases. So in the third step, again, you run it, and then you basically use softwares here, which are used for generating peak lists. That means you have run your sample completely on this, and you just have a software for pulling out all the signals that were possibly present in this are picked out. Then the software, of course, that is again an area of work where it has to be refined. It has to identify what are true signals. It has to identify what could be false positives, then what are adducts, what are isotopes, all these things it has to club and say that these are all the signals, whereas these are all the possible metabolites. So you get basically a very big list of, like it'll be a matrix, you can say, and that is what you will export into an Excel and then work further from there. And of course, the annotation is, like I said, you search against the library. So the library search is again 2 steps, maybe more advanced software will probably combine them together. But basically one is based on the accurate mass, which is the mass of the metabolite. And one is the fragmentation library. That means, again, like I said previously, when you fragment something, you get specific product ions, which is considered to be a fingerprint of that. So whether you get any matches from that or not. But you have to remember that at the end, again, everything is tentative. So unless you are able to corroborate your results with standards, you have to, talk in terms of this being like a high possibility of this being whatever you're seeing, but not 100%. So we are trying to offer like, I would say to some extent, best of both worlds, where we also run some standards, whatever is available with us. We have like a mix of maybe 20, 30 standards or something. So at least a handful of those signals which are that you pick up from this can be positively identified. So I feel that is one step. So these libraries, again, you can also build your own in-house libraries. Alternatively, you can use external libraries like a human metabolome database, or you have KEGG, or you have Metlin, and for plants, you have PlantPsych and other things like that. So then comes the next step, which is basically how to process the data. So if one objective is to basically try and find out every metabolite that is present in a biological matrix, which I feel is a tall claim and may not always be possible, because we also have to appreciate the fact that all metabolites may not be compatible with LCMS. Some may be picked up only on GCMS, some may not ionize with ESI, you may have to use something else. So all those things you have to keep in mind. And also the problem of false positives is there. So how do you basically filter that data? How do you make sense of that big sheet that you get? What is genuinely a signal? So you have to find ways to, use quality controls, you use CV values, then you filter based on some criteria. You can use single standards that can be done to refine this. And basically, you can see from the figures, these are all, heat maps and some discriminant analysis tools or volcano plots. These are all also freely available tools. Many of you might be already familiar with it. And basically, you upload those sheets here, and you're able to pull out this information. So at least at one look, you can say that if you are comparing 2 kinds of samples, like say example for a control and a disease set, whether a difference is there or not, that you can pick from here. And then finally, you have to go back further, delve deeper into it and find out that what are those metabolites which are responsible for this difference. So while in the previous analysis, in the targeted one, you can put a figure on what amount you're finding because you have a calibration curve and a standard. Here, the results are typically semi-quantitative. So you can say that this thing is absolutely absent or present in one sample versus the other. Or you can say that this is 10 times lower, 10 times higher, and so on and so forth. And then these days, the logical conclusion of this is doing what is called the pathway mapping. This is a representative figure I've put. This is actually just the tyrosine metabolism. This is what you get from KEGG. And you can see that, just as a representation, I'm saying that tyrosine is involved in several pathways. So this is where you start combining things from not just the metabolomics data, but also the transcriptomics and proteomics. You will see all the enzymes involved. So if in your result you find that tyrosine and some other related compounds are upregulated or downregulated in a specific case, then those things will get highlighted here, and you will be able to make a more informed decision on what are the pathways that are involved, which are dysregulated. So that information you can get. So that's about it. But both targeted and untargeted, they both have their issues, like they have their pluses and minuses, and one has to carefully consider while, as a biologist, you will have to figure out which is more relevant to your study. So yeah, I think that's about it, and I'm happy to take any questions if there are.
Okay, Avik Chattopadhyay has a question. I would like to ask him to please unmute himself and ask the question, please. Avik. Avik, can you please unmute yourself?
Yeah, can you hear me? No.
Yes, yes.
Yes.
Yeah, so I'm just asking, is it possible to study the metabolome of the cell culture supernatant? I mean, not the cells. So after experiment, the supernatant that is derived, can we study the metabolome from that?
Yes, so we have done such studies, but one consideration here is that typically the cell supernatant is usually, basically, it's also the cell media, right? So there'll be a lot of salts and all. So we may have to have a cleanup step. We might do some kind of reverse phase extraction or something like that. So having done that, definitely
Padma, you're missing a few
I think
sentences.
1 or 2 projects like that where we have studied supernatant.
Oh, Padma,
sorry.
you missed a few
So I'll
words.
repeat
Could
myself.
you repeat, please? Thank you.
Am I audible now?
Yeah.
Yes.
Yeah.
Okay. So Avik, I was just saying that in principle, it should be possible. The only problem with the cell supernatant is that basically it's a secretome in the media, right? And your media is expected to have a lot of salts and other stuff. So we just have to find a way to get rid of that. If you do that, it should be possible to run it. So we could also have a control where, you have just, just the media alone, and then the cell with the media, and you can also have a comparison of what are the things that have been kind of, it should be, in theory, it should be possible.
All right, yeah.
Next, I think Lakshmi M. Sundar wanted to ask a question to Padma. Lakshmi, can I ask you to please unmute yourself and place your question, please? Lakshmi?
Okay. Should I try and read it out from the Q&A?
Yeah, she has not asked her question on the chat box, actually. So, okay, there is a question. Okay. Do you require standards for metabolome-based molecule identification?
Yes. So in the targeted one, definitely we need it. And so again, it depends on what is the question you're asking. As a biologist, if you are looking at specific things, then it is definitely preferable that you arrange for a standard. Otherwise, there have been some times when, it's not possible to arrange the standards at all. So there, what is the next best thing is that, we run it on an HRMS system. And we can kind of refer to other literature where people have already worked on it. So that way we will know the masses and the expected product ions. So you can hazard a guess like that, but the best case scenario is that you double confirm it with the specific standard.
Okay. There's a question. So Lakshmi's question seems to be on the chat box now regarding plant metabolomics, especially for medicinal plants. What kind of analytic support can be availed from BLISC?
Okay, that's a very broad question. So I think so we also have HPLC system, HPLC with PDA. So again, if it is targeted, we have a couple of standards available with us. I mean, more than a couple of standards, let's say about 20, 30 phenolics and flavonoids. Apart from that, the plant Metabolome, if you may say, is huge. Like, you know, there is like secondary metabolites and there are so many things. So again, if you are looking at specific things, you can, you know, submit the standards. We can build a new method for you, or we can use the existing one if that helps. Alternatively, we can also use the untargeted workflow. I don't know if it answers it adequately, but I think if more detail is required, I can take this up offline with her individually.
Yeah, there is a specific question. Can you tell us more about direct infusion mass spec metabolomics?
Okay, so we don't do the direct infusion so much. I think in some cases it's done, but I personally feel it's always good to combine it with some kind of chromatography, because when you basically get the output of the untargeted metabolomics, you will see how it complicates things. You know, there are so many signals that are coming up. It's not like you just see one signal corresponding to the compound that is present. So chromatography is very critical. That way you're carefully able to build what are adduct peaks or isotopes and what is coming from one main thing. So I feel, and also it is complicated by the fact that you may have many isomers and things, right? So for example, like L and D cannot be separated, and even otherwise structural isomers, like all these plant secondary metabolites and many other things, they have structural isomers. They will be co-eluting. So if they are co-eluting, of course, then even by chromatography, you cannot do it. But I feel the the direct infusion thing is, we don't do it. And I personally don't feel that it's a great way of doing it, unless you have cleaned up your sample to kind of, you know, remove much of the things that you feel is not required. If that is there, then you could still consider it, but I would still prefer to go it with do it with combined it coupled with the LC.
Dr. Himanshu Behra is asking, is it possible to study metabolomics in case of ocular samples?
I think ocular is to do with the eye, is if I'm not mistaken.
Yes, yes.
Okay. So I think it should be possible, but again, in the eye, I don't know which specific compartment
is it considering the tears or the ocular tissue, Dr. Behra? You might want to clarify this, please.
Yeah, I'll put him on the speaker, okay, so that he can ask his question directly. Dr. Himanshu, can you unmute yourself, please?
Yes, ma'am. Yes, ma'am. Am I audible now?
Yes.
Yeah,
Okay.
Dr. Himanshu.
Yeah, actually, I am working on ocular samples. So my question was, is it possible to study some aspects related to this tear specimen? Means tear specimen,
Yeah.
a healthy individual as well as a patient. Suppose if
Yes.
we are looking for some diagnosis of any disease in the tear specimen, so is it possible like that we can just follow this metabolomics like here?
Yes. Definitely, you should be able to do it. I mean, I think this is, I have come across some studies which are doing that. You know, different conditions of the eye especially can be, you know, so typically the untargeted workflow can be used for these kind of things where you can hope to find some markers to pick out like early diagnostic markers of a disease or something like that. And I think it's maybe we may have to fine tune some kind of extraction specific to this matrix, which is the tear, you know, the tear component. But the analytical part largely would be the same. The whole workflow would more or less be the same. And in principle, we should be able to do that. Now, how much amount of the sample we need, those things probably will need to be optimized, but it should be possible.
Okay, means we can concentrate on healthy patient samples as well as some disease patient sample.
Definitely. You can, even within the diseased also, you can have different stages, like especially like, you know, if you're talking of cancer or something, you can bring in samples from stage 1, stage 2. So there are various studies that are designed like that also. So as you see that a specific marker is showing up, you know, in greater amounts, then so it doesn't become you don't assign a causative thing to that marker, but you can say that, yeah, this pathway may be affected. So then you go back and say that maybe this protein is affected or this gene is affected. That is how you would probably go about it.
Through real-time PCR or microRNA like that.
Sorry?
We can just analysis through the real-time PCR or microRNA for the backward analysis.
So I think, yeah, so the genomics part, of course, Dr. Avdesh is the expert on that. But I think from the same sample, if I'm not mistaken, you should be able to do metabolomics, proteomics, transcriptomics. All three should be possible from the same sample.
Okay.
If I'm not wrong, yeah.
Okay. Sure. Thank you.
So I think I will move on to Sabyasachi, but with the last question to Padma.
Yes.
Is it possible to study novel metabolite compounds if we don't have a reference related metabolite sequence for further analysis?
Okay, so yeah, that is slightly tricky, so there are a couple of software which have inbuilt in silico tools. So, you know, if you're able to capture the fragmentation, so they may not 100% say that this is this compound, but it could probably assign it to a class. So for example, if there are some product ions which belong to, say, the flavonoid category, for example, and if that's a characteristic fragment found, then you could at least say that this belongs to that. And then finally, if you want to isolate it and identify the structure, then of course it would be beyond this, where you would find a way of isolating it, you know, fractionating it, isolating it, and probably deducing the structure by NMR or something like that. But these are some in silico tools which are built into the software, which could give you an idea of what class it belongs to. That can be done.
Yeah, I think Atif, now shall we move on to Sabyasachi?
Yes, please. I think just before that, there's a general comment that I know there are many questions. So we'll try to accommodate as many possible at the end again, and then we'll record them and try to answer them offline on an email or something.
Yeah, so
I hope that works.
Before we move on, I just wanted to tell you that there are a lot of questions from the audience. Yes, we know that you are asking about the training workshops and price list for these customized experiments, as well as whether we have a recording available, whether we will be able to, you will be able to contact us. All such details will be answered. Please hold on for a while Dr. Sabyasachi speaks.
So this is the speaker, Dr. Sabyasachi Haldar, who did just a brief background about him. He did his PhD from Bose Institute, Kolkata, followed by a postdoctoral stint at University of Pennsylvania, United States. Coming back to India for work and worked with companies, biopharma companies like Hospira, Pfizer, Stelis Biopharmaceuticals, and Biocon before joining us last year. Dr. Haldar brings in the aspect of industrial, analytical capabilities and analytical requirements, and how he works to, offer solutions for a new class of drugs. Well, not new exactly, but the biologic drugs, which require a lot of extensive characterization before they are marketed. A very well-known example of a biologic is an insulin, but now we have monoclonal therapies and so on and so forth, which are large molecules and require extensive amount of characterization. And that's where Dr. Sabyasachi's expertise lies. I'll let the floor be open for him to give a quick idea about what work he does in the lab. And yeah, over to you, Sabyasachi.
Hey, thanks, Atif.
Sabya?
Yeah, so can I audible?
Yes, Sabyasachi, you are.
Yeah,
Okay.
you are audible. Please go ahead.
Okay, thanks, Atif. So, but it's a very nice introduction. Yeah, nice to hear kind of introduction where I'm really contributing something about the industry. Yeah. So what I do basically. So let me try to explain within a couple of sentences, because it's I think it's a too long discussion. Bunch of people already discussed. So basically, I'm not going to discuss about whatever the biologics and whatever, but I can just say a couple of sentences. Otherwise, maybe I cannot just introduce all the topics. Yeah. So the biologics, I mean, we may everybody know actually the biologics is a kind of the entities can be extracted from the biological entity. Yeah. So any kind of molecules, whatever it's produced in the biological system is called biologics. So there is no limitation for the insulin, no limitation for the MAPS. Yeah. So it's just a biologics, and now the biosimilar. So biosimilar, the entire world. It's not only the India. So it's focusing on to get the biosimilar. So yeah. So the purpose to get the biosimilar is a two purpose. The first purpose to make a more accessible to the people, and absolutely the cheaper version. So that's a secondary, but primary is basically the whatever the innovator molecules so far in the kind of world, it's not all accessible to the everywhere. So and India is very smart about to make this all the biosimilar. So my role is to here in the CCAMP to support the biosimilar industry for some few characterization. So characterization is a kind of the broad terminology. So I think we just do mostly the physicochemical characterization. So where the primary structure, secondary structure, and the hierarchical structure. So these three structure we mostly focus. Yeah, I think kind of here the primary structure is the intact mass peptide mapping. Yeah, so it records a lot of kind of sentences to explain all the attributes. I think some of the things already Dr. Chai explained. Secondary structure, tertiary structure, post-structural modification, impurity, and the functional. Basically, we don't do too many on that. But yeah, absolutely, we have some very small capability on this functional characterization. Yeah. So the idea is to understand the molecule. Yeah. But which stage and where they record for the characterization. So characterization record for the day one. So when you start the biologics, the industry starts from the biologics, and when the molecule goes to the market. So from the day one and to the end of the life cycle, so it's a kind of go to the market. It records for the characterization different stage and the different way and the different parameters it's required to understand the molecule. And when we have to always understand for the biosimilar, so we are mostly focused on the biosimilar. To get the idea about the biologics, novel biologics, and the biosimilar, how comparable. So our target is to always show that the fingerprint similarity, right? So to show the fingerprint similarity in the biosimilar is a big challenge. It's not always easy to show the novel molecules because both molecules are extracted from the biological system, right? So it's not a easy job to kind of show all the features, primary, secondary, tertiary, the modification, all the attributes. In fact, the bioassay also, like the functional efficacy, safety, everything is not easy to show the fingerprint matching. So it's a big challenge. And you know what happened, like just because of this, all the characterization, so far whatever the biosimilar we established and it launched to the market. So mostly characterized for the innovator also, because when innovator came to the market, it did not characterize so much. So because everywhere the biosimilar characterization, it required the novel biologics characterization. And we have to show that how the similarity from the novel biologics as well as the biosimilar. So that's a very small kind of contribution, but very important contribution. We are trying to contribute to the Indian biopharma. So for the Indian biopharma industry, we could not reach to the global market yet. Yeah, but there are some kind of very highly recommended for the regulatory body, where the regulatory agency expect bunch of the battery of the studies actually, where we have to show the novel biologics and the biosimilar is similar. And that's our service. So we basically are working together with the industry rather than to giving a service. So whatever the industry we work, so we pull them to here, the CCAMP. Sometimes we just go there. But most of the cases, like we pull them to here and we sit together. They can see our data. And so that's how it's not like we just get the samples and generate something and just give it to them. So our target is to knowledge sharing. So they yeah, I spent some very short time, not too much, 4-5 years in the industry. But I can know the pain of the industry and how the people in the industry think about to release the product actually. So it's another aspect is a timeline. So within a short time and with this stipulated timeline, you have to finish all the activity. So this is the entire kind of the approach, the philosophy we have to work with the biopharma industry, and this is activity absolutely the quality of data system, and the mass spectrometry is absolutely the major kind of technology, but it's not limited to the mass spectrometry, where I kind of told that secondary structure by CD, fluorescence, FTI. There are a bunch of the technologies are there, or physical technology. In fact, the NMR crystallography also that we have also in the experience facility. The very high-end NMR is there. This is all our technology. We're just trying to combine and to get some data and to provide to industry and allowing to industry like kind of. So this is our capability we have. This is all the data can be used for the regulatory as well to drug to like kind of come to your bench to the market. Yeah. So that's it. And I'd be happy to if someone have a question in this regard. Yeah. Thanks to Atif and Devarshini to arrange this kind of interesting discussion.
So, Sabya, if before we delve into the questions, I just wanted a roundup of from you. So
Right,
the
yeah.
idea here, and this is perhaps a general question from the audience as well.
Right.
So the idea here is that this is more of a focused characterization of, or rather a comparison between the molecules that were existing in the market, and when you are making a similar. So since you are making a similar of a biologic molecule, it's called a biosimilar.
Right.
So the work that you are doing is to establish the similarity or the extent of similarity of between the innovator molecule and the biosimilar molecule, which say, for example, a small company or a startup has made in India. Did I put it correctly?
Yeah, I mean, it's
and so the idea.
Yeah, please go ahead, Atif.
Please go ahead. Yes, please.
No, you go ahead. I just want to listen your kind of question first here.
So it's not a question. It's sort of a putting in the context. So as you mentioned that these are large molecules, they're complex, and they require a lot of techniques to characterize them and show to the regulatory agencies that these two molecules, the innovator molecule or the original molecule and the similar molecule, or if I can use the word loosely, the copy molecule, are similar. And since they have to be injected or ingested by human subjects, you need to be absolutely sure that, right? So you just don't do mass spectrometry, but you do a lot of other techniques, orthogonal techniques like NMR, fluorescence spectroscopy, and DSC and so on and so forth. So which means you have the capability to do all kind of characterizations, and then these can be extended to other molecules as well, if they may or may not be biologics. Is that correct?
See, I mean, absolutely, it can be extended to the non-biologics, but the just the technology. So all the using this NMR mass spectrometry, we are just feeding some molecule and getting some information, right? And then the main challenge comes. So it's not the generating the data, right? So after getting the data, the main challenge comes the interpretation, and then all the question answers is behind the interpretation, right? So we just spend the hours and hours to understand what is the interpretation of the data. Yeah, you rightly said. So novel biologics. So most of the what of the company already they established in the market, right? And that's they already just made some kind of copyright for 10 years or 12 years or just 20 years, yeah. But the last 5 years, basically 2015 to 25, this time. So major of the biologics, their copyright is going to be expired. So if you know actually, so all the novel molecule company, they also making the biologics, so biosimilars to just kind of launch. Because why? The number one, the purpose is when they made this the innovator molecule, it was too expensive because that time the R&D costing, the entire costing was so high, and it was not well characterized that time. They just did some kind of, so because basically they record the safety, efficacy, and absolutely the characterization. But that characterization that time may not just kind of achieve that the level, whatever is exactly the required. So now what happened? They are also like kind of the Amgen also working for the biosimilar, right? So all the other Sandoz, everybody is just working the generic. Like, why they're just making this kind of the biosimilar to produce the better, because the process, like whatever the first time they made the process 1, right? So now they're just making the process 2, process 3. In fact, the clones, everything they're just modifying. Some cases, like some modification of the proteins, it can help the yield, the amount of the expression of the protein. So they're helping the costing, everything, right? So in this way, actually, this is the approach that we enter the industry, kind of taking the approach to get a biosimilar. And biosimilar, the main approach to get the market and to spread the entire world, number one, and number two, the cheaper version, cheaper. That's another option. But most of the cases, we see that like a 70 to 80% people are ending with the biosimilar, but not too much. And every day, if you take a news on all the like kind of bulletin in the biotech companies, so they are just okay. So we are just like kind of biosimilar. Our target is to 60% of the price. So people are just kind of rat racing actually to get how much they reduce the pricing also. Yeah. So that's actually the main purpose. Yeah. Tell me.
Sure. I think we should let the audience ask the questions. Sorry to just put in a context here.
Yeah, it's actually basically it requires a long discussion.
So I think now we will ask, I think we have time for only 5 more questions.
Right.
A couple of general questions, I think Atif and Sheetal, maybe you can take. So a lot of people are asking what kind of training programs we offer. If you two could please inform the audience a bit about that.
So training program for the biologics?
No, I think this is general.
Oh, this is a general question. Okay.
Just...
So, right. No, sorry, I just missed out.
Sheetal, if you are there, would you like to take here?
Yes.
For your help put up the slide. Yeah, hi Sheetal.
Yes.
Yeah.
Hi. So, hi, I'm Sheetal. We do offer trainings in all the omics platform. We do also provide NGS trainings hands-on as well. We will be launching a hands-on training with Swiss NGS facility, as well as CCamp and Blix NGS facility in next year, September 2020, with the hands-on for genomics, whole genome, as well as 16S metagenome. We'll be launching a online bioinformatics course as well for it, most probably in February. Once the dates are on, you can visit CCamp website and get more information about it. Along with that line, we also have metabolomics hands-on training. In fact, we have hands-on training for all the mass spec based technologies, but currently due to the COVID scenario, we are going towards the online mass data analysis courses, because hands-on is not possible currently. And we do give intro about how the sample is prepared, but more focuses towards the raw data analysis and type of analysis, and how we can submit those raw data and format for the publications as well. Along with this, we if you want an in-depth hands-on training, once you come for hands-on training, CCamp is known for its the in-depth and very nice trainings, as we do a certified trainings as well. So you get, once you come to us for hands-on training, you can handle the machine yourself, you prepare the samples for yourself. And we also have the batch of only 10 people. So so each and every person has a chance to do their own hands-on trainings as well, and more focus towards individual training goes on when you have less number of people in batch. And we frequently do hands-on training thrice every quarter of the year, but currently we are doing only data analysis. So currently we just finished our metabolomic analysis last month, and proteomics data analysis course will be launched within 2 months. Again, after 1 month, we'll be having metabolomics. So just keep an eye on our CCamp website, or if you are registered to our technology platform portal, yes, you can always get an email and you can register on our training platform. We will give you, Debashini will provide you the training email IDs where you can write to us if you want a specific training. We do also provide flow cytometry training. We do also have microscopy training. we have image analysis trainings as well. So currently flow cytometry and image analysis training is going on every month. For there also we have a specific number of small groups, so we can address each and every individual personally and get them trained in these. And I would also like to point out that the fees for this involved is very minimal, and we need to charge these fees so that we, as we have most of the collaboration with the industries, and we make sure that you get a good training from this. And also, if you want any information, and we do also provide group training as well. So if you have an institute or industry who you want to have a group of 10 people or 6 people who we want to have a specific training in specific area, please write to us. We can arrange the specific training in that domain for your particular group or particular interest. So please do write us to us if you want a training in other than whatever are available with us, and we will make arrangement of special training as well. And along with these, we do also, once the COVID situation is resolved, we also do a hands-on training for individual persons. So depending on 1 week to 15 days, depending on extensive trainings. So if you have any kind of training in mind and you want to have a specific training, please do write to us and we'll try to arrange that too.
Okay, thanks, Sheetal. And one other question, a general question for both you and Atif. So a lot of people are asking, what is the cost involved in some of these customized services that we provide on Omics? Any rough, any ballpark idea that you can give us?
So usually, let me tell you that we don't have a specific list on our website because each and every project is different. So usually we charge you for the machine hours and reagent used during your projects. So we would like to talk to you once we get your project and explain you what all we can do, and we give you the workflow, how we are going to go for it. If once you are satisfied with the workflow, we'll try to then we will, according to that workflow, will make you the costing. So in case of approximately telling, all the costings are depend on if you're academic. We have a special pricing for academics and industry and startups. So depending on that, yeah, please go ahead.
I'll just show this workflow that Chaya told. And it's very important that we remember this second step, and that's the first step, consultation and understanding what is required. So while we do have standard pricing for a few things, but definitely a consultation is important to understand what is the requirement and what is the end goal. I remember one of the questions was on the data analysis, what kind of data analysis, and Chaya's answer was that there is one level of data analysis where you just do a database search and give an Excel sheet. But then there is another there's a level of analysis which entirely depends on what the end result is, and that's involved. It involves our time and resources on the software and so on and so forth, and therefore it costs more. But Sheetal, it will be good, for example, if you can give a price of, let's say, an protein ID for an academic user, and a standard curve, just as an example,
yes.
and a MySeq run for genomics.
Yes. So in case of proteomics, we just want to know a protein ID of a protein. Suppose an in-gel sample you are sending, it will cost you around 7,000 rupees with the GST, 18% GST. And with the NGS training, so we have in NGS, we have two types of pricing. One is per library preparation, where the pricing is for per sample based, the library is prepared, and for the sequencing it is per flow cell based. So the pricing you will get for that particular cell. So if you have a transcriptome analysis and it goes for 1x50 base pair analysis, and it gives you 300 million reads in one flow cell output, that will approximately cost you around 2 lakhs for sequencing. And depending on the transcriptomes, whether with this microRNA, RNA, and RNA depletion dependent library preparation, the cost will vary from 3,500 to 6,500 per sample, depending on the type of RNA library you want to prepare. So that is an approximate pricing. And Vasat's what as Atif said, once you come and communicate with us and have a detailed discussion, we'll be able to see how many samples we can accommodate in one flow cell and try to reduce the pricing as well. So once we have a discussion, detailed discussion, we'll have a ways to reduce those pricings as well.
Okay, thanks, Sheetal. I would like to now take a question from YouTube from Alka Mehra for Avadhesh. Avadhesh, are you there?
Yes. Yeah.
So Alka is asking, is it possible to sequence mRNA and non-coding RNAs together with total RNA sequencing?
Yes, it is possible for human, mouse, and rat samples. So we can deplete it. We can deplete the rRNA, and we can get the mRNA as well as non-coding RNA expression data.
Okay. Another question from Soptika Pal. Can transcriptomics be done using nanopore for bacterial samples? What are the platforms to analyze whole genome sequencing data obtained from nanopore?
Yes, we can use nanopore for bacterial transcriptome sequencing. But it is better to do it on that Illumina platform, because for any transcriptome studies, we need more number of reads to get that expression of all the genes. And in Illumina platform, that the number of reads are more. It's way more than that nanopore platform. So I will recommend to go for Illumina platform for any transcriptome studies.
Okay. Next, I think there is a general question. Are monoclonal antibodies prepared only in mouse, or can be used, some other animal model can be used? Atif?
Yeah. So there's a context on this question. So if you are talking about the context of biologics or biosimilars, biotherapeutics, they're actually not made in the animals, they're made in the cell cultures. You do a fermentation reaction and then harvest the expressed cells. So that's the state of art to get the monoclonal, therapeutic monoclonals in a fermentation. And that's where the whole idea that you have prepared an in vitro molecule using cell culture and fermentation, and then you characterize it and find whether it's functional, whether it has all the attributes that a naturally occurring monoclonal will have. But having said that, if you want to do monoclonals for R&D purpose, then of course you do it in the mouse or laboratory animals. Now there are of course a lot of models which can be used to make antibodies, not just monoclonals, including something called camelids, which can be raised in camels. You have a lot of antibodies coming from horses. So yeah, there are multiple things, but therapeutic molecules do not come from animals. They come from cell culture fermentation.
Okay.
Does that answer the question of the?
Actually, it was an anonymous attendee, most probably. So I am going to move on with the next. Can we screen amino acids? This is for Chaya. Can we screen amino acids in cell culture medium using targeted quantification? For example, specific quantification.
It's actually for Padma.
Padma. For specific quantification of steam and oxidized and reduced form.
Yeah, so amino acids, actually, we have a method already. So if you're talking of free amino acids, definitely we can do it. And cysteine, the reduced and oxidized, it's a little tricky. These molecules are always a little tricky. So we have not included it in the method that we currently have, but separately we can work on optimizing it. It can be done.
Okay. Okay, there is a question from Dr. Tina Dave. May I ask you to, this is for Chaya. I will just put her on the speaker once. Dr. Tina, may you can please unmute yourself and ask the question. Dr. Tina?
Am I audible?
Yes, please.
Yeah. Good day, ma'am. I just wanted to ask if we are providing a very raw sample and if we need to get the data from exosomes or something, will you be able to do the preparation as well?
No, I think we don't provide the extraction protocols. So if you can just extract the exosome
And then give you.
yeah, the thing is that there are very simple protein extraction protocols available. So they just use the basic TRIS, HCl, SDS combination lysis buffers. So you can just slice it up. Because the thing is that if you extract at your end and you know that protein is fine, and if you send it to us, then there is a less chances of any error happening. Yeah.
Okay.
So if I may add here, this is a general question that we get a lot of times for all the facilities as to why we do not do sample preparation or extraction. The reason is simple. These are core facilities where there are variety of samples that come to us, and sample extraction and preparation itself is an art, if I can say. So we do have standardized methods for extraction for certain amount for certain sample types, but it's always best and recommended that you do the extraction in your facility, in your laboratory. Of course, we are available for a consultation and checking with us what is the most appropriate way to extract a sample or prepare a sample. For example, for phosphoproteome kind of analysis, you require to enrich the phosphoproteomes before even extracting. Likewise, to take an example from metabolomics, if you are doing untargeted, and Padma alluded to that just a simple mixing of little bit of water to methanol can change things. So talk to us beforehand. We will let you know what is, in our understanding, the best way to go about extracting the samples. Do the extraction at your end or sample preparation, and then please send it to us. Same goes for genomics, where the library is prepared by us. There's a difference here. The library is prepared by us, but the extraction is done at the user's end. Am I correct, Avdesh?
Yes, yes. Yeah.
Yeah. So extraction, as you would say, somebody is using a fecal extract and another person is using a exome sequence. So from for example, let's say just say cell culture, they're two very different original samples to begin with. So, and there could be very different upstream requirements. So it's better that you do it. This for general. I hope this similar questions are addressed by this.
Yes. So another, going back to the similar things, people who are wanting to know if there is a recording of this session going on, there is a recording. At the same time, it is on YouTube. You can access it on CCAM Facebook page, as well as CCAM YouTube and as well as Blisk YouTube. You can access it anytime you want. I think we will try to close the session now. With the last question from Rahul Krishnan. Does CCAM offer histone post-translational modification analysis?
That'll be Chaya, you.
Yeah. Debashni, may I know the name of the person?
Yeah. Do you want to speak to him? Maybe I.
Yeah, definitely. Yeah, I have a few questions.
Yeah. Rahul, can you please unmute yourself and ask Chaya?
Hello.
Yeah. Hello, Rahul.
Am I audible?
Yes, yes, you are.
I'm studying on histone post-translation modification, generally on basis of epigenetic aspects. So I wanted to know whether CCAM offer the service for doing the histone post-translation modification. I'm doing it based on cell culture, cell cultured cells.
Cell culture cells. Okay. Histone modification, I think you can do it as much as I know. There are literature. So what you need to do is you need to, the first thing is you need to narrow down what post-translation modification you are need to focus. For example, I guess in histone it's acetylation. Am I right?
So actually, I wanted to know in a general aspect. So what are the modification coming in histones after? So I study with viral infection, so I wanted to know.
See, if you see, there are quite a lot of modifications. They are present. Histone, most of the people they study as much as my understanding is acetylation, methylation, and few of them phosphorylation. And so they are the main people.
Just focusing on acetylation here.
Yeah, if you're focusing on acetylation, you can do that.
Okay, fine.
Yeah.
Okay. I think, Atif, with that, shall we close today's session?
Yeah, so General, again, saw a couple of questions on how to reach out to the colleagues for detailed discussion. I think the best way is to first drop in a line to this email ID that is perhaps on your screen right now. It's services@ccamp.res.in. One of us will reach to you and we'll try and answer it as much as possible on email, but definitely would love to speak to you over a phone call or a video call. So while we will, as a follow-up, perhaps Debashini, we can share the contacts of our colleagues, but this is the basic, most common, easiest way to reach out to us, right? And then, of course, I'll request to keep an eye on the website and the social media handles. Yeah. But as Debashini said, let's thank the speakers for taking time out. And 2 hours from a lab is difficult to manage. But thank you guys for taking a time out, explaining and engaging with about 180 people on the Zoom call itself and all other places. Thank you so much all for joining in for today's workshop with us. And Debashini, it's over to you. Thank you from my side.
So I just wanted to say that thanks for the great response from the audience as well. I mean, we had a very interesting session with very insightful discussions from all the experts from Atif and Sheetal and Sabyasachi, Padma, Chaya and Avadesh. And there were very interesting questions. Please, we will send out a follow-up mail where you will get all the email IDs and all the links that you might require later on. Feel free, as Atif said, to please get in touch with us, and then we can take the discussions forward. And I think with that, we will, I also wanted to thank Chandrakant and Mahin here from the BlissComms office for, sorry, for helping coordinate today's session. So I think with that, we will close, and thanks everybody. And see you for the next session of CTIS coming soon. Yeah.
Last में क्या बोलूं?
Thank you. I'm closing the call now.